Right, so luckily we're running past time. It's not going to take very long. I'll just give you a quick history of this project. It started in 2018. This Jamie Duckers is he's a clinician in Cardiff who runs a cystic fibrosis clinic, and he was quite interested in starting to look at the brain, and you'll see why as we go through this talk. Him and Richard Wise got a grant to start studying these people. Then Richard left Cardiff, went to Italy. He gave it to Claudia Metzer-Badley, who started running the project. And then in 2020, something happened in the world where people with a lung condition didn't want to leave their house. And so this just dragged on and on and on. And so Hannah eventually got the data to analyze it. And I was put on at the very last minute. You can see I'm 5th of 8 authors here. The second author is sitting up there. For some reason, he couldn't do it. So I'm the least qualified person in this list to actually talk about this. But I'll give it a go. So strap in. This is everything I need to know. I know about cystic fibrosis, so please don't ask many questions. So what is cystic fibrosis? Right, so most people think of it as a lung condition, and yes, it is. It's predominantly a lung condition, but it's actually about this mutation on a gene, the cystic fibrosis transmembrane conductance gene. And what that does is there's chloride channels on the endothelium of cells, and essentially it stops the chloride going in and out of the cell, which you end up then with sodium chloride, which is salt essentially and a buildup of salt in all interstitial fluids in the body. Right now, obviously in the lungs, this like turns up as a mucus in the lungs, and people struggle to breathe, etc. But it actually is everywhere else, and like it's in the liver, and they have a lot of liver conditions, and so a lot in the pancreas. You see it in the bowels, etc. And you know, people struggle to essentially metabolize food because they have too much mucus in their bowels, and etc. So it's all over the body. Yeah, increased sodium, reduced water absorption, and mucus everywhere, basically. Back in, it was discovered like in 1938, and in the 50s, you wouldn't live beyond a year. Had it in the 70s, and it was about 8 years old was the average survival rate. And since then, it just increased because they basically got better at giving people diets and things to deal with this. But then what happened about 10 years ago, these really new drugs came out, these modulators, which essentially cure it in some way, right? And they increased the survival rate to 45 years. Now, if we came up with a drug that increase someone's rate by 45 years, would definitely take the rest of the day off. You know? And so these people now, they expect anyone who's born with it now will live into their 80s. Okay, so this has gone from being a lung-based condition that's going to kill you quite early into now an aging condition. And now all the CF like funders and that are actually quite worried, because basically you're seeing all these age-related problems happening, which you wouldn't have saw before because people died before. So, what other comorbidities do they have? Interesting, like when you get to your 30s, you normally have like 5 different comorbidities as well as the cystic fibrosis. So it's a big problem. But a lot of them are actually related to cardiovascular disease. Hypertension is quite an obvious one. And they have a type of diabetes where 50% of them have this diabetes that's actually directly related to the CF gene. And it's to do with a reduction in insulin production. And so that is obviously going to have some vascular effect. And then arterial stiffness, they've got increased arterial stiffness, but even in childhood, so at the age of 12, you have a 20% increase in arterial stiffness. So to me, and the reason I'm interested in it, I think it's like an advanced arterial aging that we should be interested in. And then obviously then people then start having problems with memory function and brain fog, etc. So they may actually be related to all these mechanisms that I'm was talking about, like related to dementia, etc. So this is why we're interested in it. Lots of people are interested now in the cardiovascular stuff, but I think brain cerebrovascular function is something that we should probably be thinking of. There's like, in terms of numbers, it's actually quite, there's only about 70,000 people have it. 30,000 of them are in the US. They're mainly white, so it's a mainly predominantly Northern European ancestry that have it. But there's 1,000 people every year are being diagnosed with this. So this will actually become a much bigger group over time. So background, there's been one brain imaging study, and hopefully none of the authors are here, but like if you look at that map, I wouldn't trust any of the results here. Basically, they were looking at structural changes. They said that these blobs were changes in cortical thickness, but anything. But if you look at other studies in related conditions like pulmonary fibrosis and COPD, which is another lung problem, you get alterations in blood flow, and it's probably related to things like hypoxia and also then arterial stiffness again and cardiovascular disease. So in this was basically a pilot study, an opportunistic kind of study just to see what we could be. And so we got 14 people with CF, PWCF, and 56 healthy age-matched controls. And actually, as an aside, these came from a database that we've made in Cuba called the WANT study. So it's the Welsh Advanced Neuroimaging Database. Is that right? Yeah. And it actually has like 160 people who have 20 hours of imaging all over 3T, 70 meg, and there's lots of cognitive stuff. We've done diffusion, we've done physiology type things, lots of different data. It's all downloadable. It's just being released actually at the database. So if you need health controls, please go get them. And we use propensity matching to actually try and get the most amount of the healthy controls we could to match the CF group. And I basically looked at empty range, got Picasso, got the trust times trust sequence, and then we did a T1 inversion recovery to calculate hematocrit or the hemoglobin levels. And if anyone wants to know about that, Mike was the one who kind of developed that idea, but it's quite good for we used it quite a lot to adjust for different hematocrit levels. And then we just did simple battle, estimated CBF, and then used the global measure or the gray matter measure of CBF and the OEF from the trust to give us a global CMRO2 measure. I know there's some issues with doing that because that doesn't brain the whole brain, etc. It's like mismatch between what you're doing, but it seems to be a good proxy of global CMRO2. So the results basically, people are the same intracranial volume, but you actually do get a change in global cortical thickness. It's quite interesting. But surface area is the same though. And then in terms of physiology, we were expecting a CBF change. We didn't really see one, not a significant one anyway. And the OEF wasn't significantly different, but then when you actually try and calculate this global CMRO2, you do get a significant difference. So it seems to be something happening. I think I would assume the more people we put into this, if we get more people, we would actually see a CBF difference. And this is kind of what we're interested in. If you look at the regional CBFs, you actually do see some areas. This is the right-hand side here is FDR corrected CBF areas, and this is just kind of a list of them. And the ones that you actually see a statistical difference, and a lot of these areas are related to cognitive function, memory, and emotional regulation, things like that. So it seems like there may be a difference in CBF in these areas causing these memory problems. So that is kind of like what the study was. Apologies, these slides are really wordy. It was because Hannah thought I wouldn't know what to say. She's right. But yeah, I basically said everything here, I think. But we're interested in doing another study. We want to do the dual calibrated MRI stuff to actually look at CMRO2 and other regional differences in OEF CMRO2. And we've kind of done this sort of stuff in other patient groups, such as Scanlon's done a multiple sclerosis one, so I'm going to take the same approach. And one thing that Mike has developed is looking at mitochondrial partial pressure of oxygen at mitochondrial level, which might give us an idea as to why you would see a difference. And there's like some idea that there could be altered mitochondrial function within these patient group, and so maybe we can try and get to that type of modeling. So the new follow-up study, we're going to start this next year. I'm really interested in the arterial stiffness thing, and basically what Jakob was talking about at the start of the week, how the stiffness actually progressed through the brain, and what knock-on effects does it have then on OEF02 and basic damage. And we're going to try and use Einstein's webcast to tell us brain barrier function, etc., which was presented earlier as well. Extend the group, and we're trying to exclude them for the cardiovascular disease, just so that we can see if there's anything related to CF. And then the open questions are: we want to do these CO2 in these patients. The people we scanned have good lung function, but obviously, it's hard to know whether they will tolerate the CO2 or not. So we'll see. The clinician that we're working with seems gung ho. He seems happy to do it like that. So I guess we should. Some questions then about like CBF, and do we have to worry about like arterial oil, etc. And then like we need to then decide on our DIMAC sequence that Ian showed about looking at pulse and pulse wave velocity, where to look and what we're going to target. That's pretty much it. Thank you.